The next part, well, let's welcome Professor Huang Liming, who is a superintendent of Taiwan Medical University and the Children's Hospital. And let's welcome Professor Huang Liming. Thank you.
Good morning. So I'd like to introduce the next speaker. The next speaker actually appeared in the previous slide. You saw his pictures. The speaker is Charles Chiu. I think in Chinese it's Chiu, maybe. He's an ID specialist by training. He got his MD degree in 2001 in UCLA. And then he got his PhD degree also in UCLA in the field of biophysics. After that, he had finished resident training in internal medicine, and then do his ID fellow training. And so now he's a board certified ID specialist. His position currently is a Professor Laboratory Medicine, Division of Infectious Disease, UCSF School of Medicine. And he is the director of UCSF Abbott Viral Diagnostic and Discovery Center, also associate director, UCSF Clinical Microbiology Laboratory. So from his experience, you know he's very qualified in this field of using the new diagnostic technique to identify potential pathogen. I heard his talk last year, which was very impressive. So now let's welcome Professor Chiu.
Thank you, and I would like to thank the organizers for inviting me to speak with you today. I did speak last year. This year's talk is going to be updated. I should also mention that my name is Chiu Huayan, Zonghua Mingguo de Huo. So if you can speak to me in either Chinese or English, I prefer to speak English. So I'll be talking to you today about clinical metagenomic and host response sequencing for diagnosis of infections. So just have a few disclosures. I do get research support from Abbott Laboratories. I'm on the scientific advisory board for 3 companies. And I also have patented the software that's used for pathogen detection from next generation sequencing. So this is a slide that you may have seen last year, but I just want to introduce this as a way to show kind of the situation for us as infectious disease clinicians. There are many current challenges to diagnosing infections. One is that the diagnostic yield tends to be still poor in many acute infections, whether it's meningoencephalitis, pneumonia, or sepsis. And this is despite all available diagnostic testing. The typical infectious disease patient undergoes multiple tests that are done serially without really yielding a diagnosis in many cases. And as a result, many patients can be in the hospital for days to weeks. And this ultimately contributes to prolonged hospitalization and progressively invasive testing that's required, as well as unnecessary treatments. In addition, there are huge costs to having these patients in the hospital without a diagnosis, and it's a huge healthcare burden to the healthcare system. And so our approach is, and this is was also been described by Dr. Wilson, our approach is to apply metagenomic sequencing as a single test that could potentially identify nearly all infections by detecting specific sequences. And so this is the idea, is that we can implement clinical metagenomic sequencing in the setting of a CLIA validated licensed clinical laboratory, such as what we have at UCSF, as well as in many hospitals. And the idea here is that we would run the test and be able to couple that with automated bioinformatics to be able to yield and provide a clinical diagnosis. Ultimately, the goal of this is to really provide cost-effective and actionable information that could be used to guide treatment of these patients with critical illnesses that would improve patient outcomes and lower healthcare costs. And really importantly, with respect to the turnaround time for this assay, it's important that the assay be done in hours to days versus days to weeks, because these patients are critically ill. Many of them can decompensate and become worse within hours. So it's really important to try to achieve a reasonable turnaround time for these types of assays. So we've developed and validated these at UCSF, and we currently offer both a spinal fluid assay, and of which Dr. Wilson has talked about in the previous talk, as well as an assay from plasma. And I'll also talk about some new assays that are currently in development. And what we typically do with the assay is, you can think of the analysis of the sequencing data as a black box. So we basically, this is run in entirely automated fashion. Typically, we generate about 50 to 100 million sequences per patient sample. And the data is analyzed actually on the cloud. We have a cloud computing system. And the results are provided for the clinical lab director to sign out essentially twice a week or whenever the test is run. So the idea behind this is that this is an entirely automated process, and in that way we can analyze more samples in a timely fashion. So the clinical utility of this was demonstrated in an article that was published by Dr. Wilson and myself and others at UCSF, titled "Clinical Metagenomic Sequencing for Diagnosis of Meningitis and Encephalitis." And many of you may already be familiar with this study. I just want to simply highlight that a key finding from the study was that roughly a quarter of all infectious disease diagnoses were made using metagenomic sequencing alone. And this is really important because it suggested that metagenomic sequencing as a test could be complementary to existing tests, and it would result in increased diagnostic yield for these difficult to diagnose patients. And since then, I show you this slide here just to give you some idea of the positive cases that we've been able to identify over the past 5 or 6 years. You can see here that they really span the whole gamut of different viruses, bacteria, fungi, and parasites that cause infections. Several of them are rare and uncommon infections. Many of them, there really are not diagnostic tests that are readily available. Most of them have to be sent to specialized reference laboratories for testing. And I also want to highlight one example, and this was also I talked about it previously. There was one example of a patient who presented with a hepatitis E neurological infection, meningoencephalitis. And at the time that she presented, she was had developed fulminant liver failure, was on the liver transplant list. But because we were able to make the diagnosis using metagenomic sequencing, we were able to help her avoid getting the liver transplant. She was treated successfully with ribavirin. I also want to highlight another example where this is a case of a vaccine-associated rubella encephalitis. So we not only were able to diagnose rubella encephalitis in this patient who was a young girl, but we were also able to do phylogenetic analysis to analyze the sequence, and we were able to show that, in fact, that she had been infected by essentially the viral component, which is found in the rubella vaccine, which is a live virus. So she actually had received the rubella virus kind of earlier and was later on found to be immunocompromised. But the idea behind this is that we were able to not only leverage metagenomic sequencing for detection, we were also able to use it to sequence the rubella virus and to place it phylogenetically on the tree. I want to give you another example. This is an interesting case of a 70-year-old gentleman who presented with a chronic meningoencephalitis. He actually had symptoms that had persisted for 5 months. He lived in the rural Midwest and actually had been bitten by mosquitoes for, actually had received, his wife basically said that he had hundreds of mosquito bites. And what we detected, interestingly enough, was an unusual virus that most of you, in fact, probably nearly all of you may not have heard of, which is called Potosi virus. And part of the reason why you may not have heard of this virus is it's a virus in the Bunya virus family, but human infections from Potosi virus have never been described. So it really illustrates another potential use of or utility for the test, which is in identifying emerging viral infections or perhaps a novel or unexpected viral infections. I want to move really quickly into kind of some of the new work that we're doing. One is that we're very interested in doing metagenomic sequencing of body fluids for infected patients. And the idea behind this is that what applies for spinal fluid, in principle, the same metagenomic sequencing assay or technique could be applied for many other body fluids. So this includes pleural fluid, peritoneal fluid, abscess fluid, joint fluid, BL, and urine, as well as spinal fluid. And so this was work that was spearheaded by a very talented fellow at the time, Dr. Wei Gu, at UCSF. He's currently on faculty at Stanford. But the idea behind this is that we wanted to apply the metagenomic sequencing test really to a wide variety of different body fluids and be able to validate that. And so I want to give you a couple of examples of how it applies. So this is a proof of concept where we simply applied the assay on a respiratory sample from a patient with pneumonia. Now, we knew that this patient was actually infected with SARS coronavirus 2, but we wanted to test, in principle, could our assay have detected COVID before there was a sequence available. And indeed, we can detect it because the top hit actually is that this is a bat SARS-like coronavirus, WIV1, which stands for the Wuhan Institute of Virology in Wuhan, China. And so, and at the time, that was actually the closest relative to SARS coronavirus 2 because the genome of SARS-CoV-2 had not actually been submitted to the database at the time we generated the database for SERPI. Now, once you actually update the pipeline to include the database, now you indeed get the top hit being severe acute respiratory syndrome, coronavirus 2, or SARS-CoV-2. I want to go into another example. We've been working on an assay that we now have clinically available, which is an assay looking at blood. And so this is a plasma assay, and there are several potential clinical indications: culture-negative endocarditis, fever of unknown origin, perhaps difficult to biopsy and organ disease, and also immunocompromised and transplant recipients who may have suspected infections, as well as ICU patients who are on empiric antibiotics. And so this is a case of a 33-year-old female who presented to the hospital with sepsis and pneumonia. She had 2 weeks of flu-like symptoms, cough, fever, and chills, and sputum production. And it had been transferred from the outside hospital with respiratory failure, was started empirically on antibiotics, including antifungal medications. Did have a procalcitonin level, which was 8. Which was normal, but it was still still unclear exactly what was causing her symptoms and her disease. I should mention that all microbiological testing ended up being negative, including 4 sets of blood cultures. And she actually had some risk factors that were potentially suggested a wide variety of potential infections. One is that she lives in a cabin. She had no history of recent travel, although had traveled to Mexico as a teenager. Her boyfriend had become ill with a flu-like illness about 2 weeks ago, prior to when she became ill, but had recovered, and no diagnosis was made in his case. And in addition, the patient stated that she was hugged by a homeless woman who was hospitalized for methicillin-resistant Staphylococcus aureus infection, or MRSA. And then finally, she had unusual exposures to dogs and cats and wild turkeys, as well as a rat infestation in her boyfriend's house. So really, the differential diagnosis was quite large. And this is not an uncommon case a very complex case where the history would suggest potentially wide variety of different organisms where we really lack kind of diagnostic testing, which is primarily based on looking for one organism at a time. So we felt that this was actually a very interesting case to actually address through plasma sequencing. And I can show you a few images. This is basically her kind of chest x-ray showing kind of the upper lobe infiltrates that were observed, as well as the tree and bud distribution and the infiltrates observed on chest CT scanning as well. Ultimately, the metagenomic sequencing showed that this was a case of Haemophilus influenza pneumonia and sepsis that was culture negative. And so we did metagenomic sequencing of both the bronchoalveolar lavage as well as blood, and we were able to detect Haemophilus influenza about 2 to 3 log higher in the BAL fluid. And what was quite interesting was that after the sequencing results were available, 1 of 4 cultures from the outside hospital was actually found to be positive for Haemophilus influenza. But what was it was actually ended up being dismissed as a contaminant. And unfortunately, the clinicians at the other hospital, at the laboratory, didn't actually let us know that one of the 4 cultures had turned positive. And this was kind of after our sequencing results were readily available. Eventually, we did confirmatory 16S universal PCR from the bronchial lavage fluid that confirmed that Haemophilus influenza was indeed the diagnosis. But what was really unusual about this case was that this was a 30-year-old woman who had no past medical history of any disease. So why would she suddenly develop critical illness from Haemophilus influenza? I mean, she's otherwise healthy, really has no history of pneumonia or other sicknesses in life, no history of any immunocompromised state. So we wanted to do some further investigation, and that was also done. The simultaneous workup was to see, could there be some underlying reason why she had developed the sepsis? And so this introduces kind of an interesting paper that we actually published last year, which was titled Detection of Cryptogenic Malignancies from mNGS whole genome sequencing. And the idea behind this, I don't have time to actually go through the paper itself, but the idea behind this, and you're free to read it, but the idea behind this is that we're able to look at the DNA metagenomic data and be able to identify copy number variation, essentially chromosomal variation in representation directly from the DNA metagenomic data. And by doing so, it enables us to identify potentially some translocations or other signs of cancer that are typically done using cytology. And so we wanted to actually, so while the workup was being done for this patient, we also wanted to kind of run this new analysis. And indeed, we were able to show that this was actually that she did have acute myeloid leukemia, and this was confirmed both by a biopsy as well as cytology, and she had kind of monosomy 7 and pleural fluid, which basically, and it was probably her underlying leukemia that had predisposed her to developing Haemophilus influenza, sepsis, and pneumonia. But it really illustrates kind of the potential utility of the assay in that potentially you may not only be able to identify viruses and bacteria or diagnose infections, but you can actually look for some of the hallmark cardinal signs of cancer, perhaps through using this tool as a liquid biopsy test. I want to move into a little bit of some of our work with nanopore sequencing. So we're continuing to do some interesting work in nanopore sequencing. I want to say that we've now developed essentially a method of going directly from the sample as opposed to doing extraction. So the idea behind this is that we're able to say, collect a sample, say a respiratory sample, inactivate it, and prepare the library within an hour and then put it on the sequencer. So the idea is that the turnaround time for a test such as this would be 2 to 3 hours versus typically 24 hours for most metagenomic tests. And so you might imagine that there are potential applications to this approach. We're currently using it for SARS coronavirus 2 sequencing, but there are other applications, including potentially diagnosing respiratory viral infections, diagnosis of body fluids, et cetera. And I'd be happy to talk to you a little more, but that's one area that we're working in. I want to also move into kind of this other huge area, which is looking at host response. And I know that Dr. Wilson has talked quite a bit about it. I'll be focused on using the diagnosis for host response kind of broadly across various categories. So our idea behind this is that we now have data from roughly 4,000 spinal fluid samples that have been collected and representing a wide variety of different bacterial, fungal, parasitic, and viral infections. But in addition to that, through a collaboration with Dr. Wilson, we have samples from patients with autoimmune disease as well and other non-infectious etiologies. And these are all spinal fluid samples. And our idea is that because we do RNA sequencing to identify RNA viruses, why not take the same RNA sequence data and be able to then use it to diagnose other infections on the basis of the human host response. So the general rationale for this is that these infections can potentially be quite distinct because a patient will have a unique or distinct signature in response to the infection, or a unique or distinct response to that particular infection. And can we potentially leverage the RNA data that we're getting to be able to generate models that can predict whether or not a patient has an infection? Now, you might envision that this may have quite a bit of clinical utility. For instance, not uncommonly in infectious diseases, we encounter cases of bacterial positive cultures where we think it's likely a contaminant. So for instance, in blood culturing, we often can culture, say, Staphylococcus epidermidis, a coagulase negative Staphylococcus, or potentially culture P. acnes. And we may struggle to determine, is that really an causing infection or is it a contaminant? So one way we can sort of address this potentially is to look at the host response. So the idea behind this is that what we do is we take roughly 70 to 80% of the data, we then develop it as a training set, and then we train and develop a model that can be used to make predictions on what kind of infection or what kind of illness it is. And then we apply the model on an independent test set, and we use the results of the test set testing to be able to evaluate the performance of that model. So this is another way of looking at it. And so what we typically do is we divide up the model into various binary classifiers. And I say binary because you're making a comparison of one versus the other. So in this case, we might have a classifier, for instance, that can distinguish bacterial infection from everything else, or viral infection from everything else, autoimmune disease from everything else, fungal infection from everything else, et cetera. And the idea is to accumulate or collect a whole large number of these classifiers and put all of those classifiers into a random forest classifier. So this is a kind of a machine learning based approach, and to be able to make the predictions. And so this is essentially all of the kind of gory detail that you'll kind of see here. But the general idea is we generate these classifiers that can distinguish between, say, one condition and another. And by doing so, we have the machine, we use machine learning, we have the computer select the genes that would represent, for instance, bacterial infection or fungal infection or parasitic disease. And the random forest classifier, the consensus classifier, will take all of that information and be able to basically produce a result or a prediction as to what is most likely causing this particular illness based on the patient's host response. So, how does this actually work? I want to show you some examples here. So an example is like a subthreshold result. By subthreshold, I mean a result that technically is negative because the number of reads we detected was not above a predetermined threshold. So we had a case of a subthreshold result for Mycobacterium tuberculosis, as Dr. Wilson said, a very important cause of meningitis. And you can see here that the mNGS result was negative because it was subthreshold, but we were able to analyze the host response from the same sample, the same data, to predict that this is a bacterial infection. We also had a case of a GABA receptor, and this is an autoantibody receptor-mediated encephalitis. And you can see here that the metagenomic sequencing result was negative, as you might expect, because this is an autoimmune disease. And indeed, this is concordant with the host response. We had another case where we had a sample from a patient who likely had a viral or autoimmune disease, but the mNGS results showed that there were actually multiple bacterial species present in the sample. But if you look at the identity of those species, it's most likely, it was thought that they were most likely due to contamination. And indeed, the although we never really got a diagnosis for this patient, it was actually concluded that the patient probably had a viral infection. The host response did suggest that it's more likely viral. We had a case of a subthreshold Aspergillus infection, which is a fungal infection that's very hard to diagnose. And you can see here that we're able to call it as a fungal infection. And then finally, I want to introduce, we had a case of a Zika virus infection, which was quite interesting because Zika virus, although it's a viral infection, it was not actually represented in the model. In other words, there were no cases of Zika virus meningoencephalitis in the training set. And that's because this is considered a one-off sample or an outlier because we only have like one case. And so, but it really illustrates the point that potentially you may be able to identify other types of infections, even those that are not specifically represented in the training set data. And so this is an ROC, or receiver operated characteristic curve, showing you the performance of the classifier. You can see that it's actually pretty good for most of the categories except for parasitic infection. I suspect that part of the reason why parasitic infection does not perform as well is because we have many fewer cases. We only have about 10 to 15 cases actually in the data set. But I want to give you an example of how we would potentially use it to diagnose parasitic infection, because we have cases like this where it may potentially be needed. So this was a couple who went on honeymoon in Maui in Hawaii and who had returned to the US, but then both of them developed at the same time fever, rash, headache, and neck stiffness. And workup showed that the couple had actually developed cerebrospinal fluid eosinophilia, which is a hallmark of parasitic infections, although you can also see it with other conditions. And upon questioning, they stated that they were hiking in the jungle and picking up and eating raw fruits. So some of you may be aware that in Hawaii, there is a parasite which is circulating there, which is called Angiostrongylus. And Angiostrongylus has a very interesting life cycle, which involves partly in snails. But to make a long story short, we were able to identify this as Angiostrongylus. So this is kind of our SERPI pipeline. You can see that the cases from the couple are the first and the third columns. And you can see here that we detected reads to Angiostrongylus cantonensis, which is a worm which actually causes this disease, eosinophilic meningitis. But what's quite interesting is that, as I said before, the parasitic classifier does not behave very well. And in fact, it was negative by our classifier. But we've actually developed sort of a different type of classification algorithm called based on rank correlation. So the idea behind this is that we have all of these different types of infections and all of these types of non-infectious etiologies. So what we potentially can do is we can take that clinical sample and directly compare it based on a limited subset of genes to every other sample in the database. And the idea is that if there is a sample or a host response that's related to the patient, it would be able to detect it. And in fact, when we did this, on 300 samples in a random comparison set, you can see that the husband, basically, his sample actually was paired with the wife sample, where the wife sample was actually number 3, was actually third. So it could be viewed as potentially quite significant because it's number 3 out of 300. But in addition to that, we were also able to see other parasitic cases that were kind of high up on the list, like Taenia solium, for instance. So what we've now done is we've actually developed an algorithm, again, using machine learning, as a way to basically predict kind of the likelihood of having a parasitic infection. And what we're viewing ultimately for the model is it would not only use the classifier that we previously developed, but also use this kind of subcategorization using rank correlation coefficients as a way to specifically identify certain diagnoses. We've applied it, for instance, on Mycobacterium tuberculosis, and it appears to work quite well for that, as well as other infections such as Lyme disease. And so this is kind of one way of looking at it. This is called an LDA plot, which stands for linear discriminant analysis plot. It's essentially very similar to or analogous to a PCA plot, a principal component analysis plot. And the idea behind this is you can see at a glance, and this is plotting every single symbol here is a patient or a patient sample. And you can see here that, as you might expect, there's quite a bit of overlap between, say, a viral infection and an autoimmune disease, which is not unexpected, as well as a parasitic infection and a non-infectious autoimmune condition. But the other categories like bacteria and fungi are more discrete or spread out. I want to move into another application of host response using metagenomic sequencing, which is really for Lyme disease. And so I want to introduce the fact that, or kind of the challenge that Lyme disease is very difficult to diagnose for us clinically. Lyme disease really consists of 2 phases. One is kind of an early phase, where a patient who is shortly after being bitten by a tick presents with fever and a rash. And then there's a late phase where patients may develop musculoskeletal and neurologic symptoms. And although there are no cases of Lyme in Taiwan, I did check that there have been cases diagnosed in Taiwan. Essentially, they have all been imported cases, but this is still a big problem, especially in the United States and Europe. In the United States, for instance, we have an estimated, the CDC estimates anywhere from 500,000 to 1 million cases of Lyme disease a year. And so the fact is that we really lack both an accurate or sensitive test, both for acute infection as well as for chronic infection. So when we think about Lyme disease in terms of tick-borne infections, also it's very difficult to diagnose because there are a wide variety of tick-borne infections that can cause a very similar clinical presentation. So what we've done is we've developed, essentially using RNA sequencing or metagenomic sequencing, we've developed actually profiles that can help predict, for instance, different types of tick-borne infections, as well as other infections that may mimic these febrile infections. And the other thing that we also noted is that there's a persistent gene expression signature in Lyme disease. So if you look in, say, visit 1 at the time of presentation, there's a very strong inflammatory signature, but the signature is actually persistent even 3 weeks later. And so we've leveraged this into a paper which we just published actually about a month ago, showing that we've developed a host response classifier with really excellent performance for diagnosis of early Lyme disease, 90% sensitivity, 100% specificity, and 95% accuracy. And notably, there are several positives were actually in patients who actually were antibody negative, but were thought clinically to have Lyme disease. The other thing that's also quite interesting, and this is what we're working in collaboration with investigators in Johns Hopkins, is that we're really interested in knowing, can this test be used for chronic disease or patients who have persistent symptoms from Lyme disease? So this is an area under active research in my lab. I want to end by talking just one last example, because I think that this particular approach using metagenomic sequencing has implications for public health as well. So there was an investigation of pediatric hepatitis cases that we did in collaboration with the US Centers for Disease Control and Prevention. Last year, there were clinicians at a children's hospital in Alabama who had identified pediatric patients, children who had severe hepatitis. And they also found that many of these children had adenovirus viremia, actually also had adenovirus in their blood. So there was always this idea that adenovirus potentially may be kind of an emerging cause of hepatitis in these children. And in several children, it was quite severe. 2 of the children, for instance, needed to get a liver transplant because of this disease, and otherwise were prior to becoming sick, were completely healthy. So what we did here was that we recently released as a preprint a paper which shows that we've detected independently from another group the association of adeno-associated virus type 2 in children in the United States with acute severe hepatitis. And adeno-associated virus is quite interesting because it's never been linked to disease. It's actually commonly been leveraged as a gene therapy vector, and it's never been. It infects readily both children and adults, but it's never been linked to disease up until this point. And so we were able to look at pediatric hepatitis cases from 6 states, as you can see here, and looking at a variety of samples: whole blood, plasma, and nasal swabs, and in a few cases, stool as well. Most of our findings are directly from whole blood, for which we had the most samples. And we applied a battery of different molecular and sequencing-based analyses. I want to just sort of like the take-home message of this is that we found in 12 of 13 cases, we found adeno-associated virus 2. But in addition to that, we not only found AAV2, but we also found several helper viruses that caused essentially co-infections. So by different strains of adenovirus, human adenoviruses, EBV, Epstein-Barr virus, as well as CMV and HHV6 in a subset of samples. And so if you look at this adenovirus AAV2, it's not only adenovirus associated 2, but it happens to be one specific cluster, as you can see here, of adeno-associated virus 2. So they were all clustered together, sort of as you can see here, both on nucleotide and amino acid alignments. And this is actually quite interesting because around the same time, actually, in fact, earlier than us, the UK, the United Kingdom, a group from the United Kingdom had actually reported adeno-associated virus 2 infection in children who had pediatric hepatitis. And when we actually compared the sequences that they have obtained from their adeno-associated viruses, they're essentially identical to the sequences that we found in 6 different states. So there does appear to be some link between the fact that we're finding the same viral strain of adeno-associated virus 2 on 2 different continents. And currently work is ongoing to try to detect, to prove an association. It's probably going to require both animal models as well as kind of more epidemiologic work. So I want to end by thanking all the members of my lab shown there on the left who actually did all of this work. I have had a long history of productive collaboration with Dr. Michael Wilson, others at UC San Francisco, as well as the UCSF Clinical Microbiology Laboratory, who have been instrumental in setting up the clinical assays. I would also like to acknowledge Lyme Disease, Bay Area Lyme Disease Foundation, the CDC, Cleveland Clinic, Johns Hopkins University, as well as other collaborators at the California Department of Public Health. And then finally, I'd like to acknowledge funding from the NIH, the U.S. Centers for Disease Control, the Sandler Program, and the Stephen and Alexandra Cohen Foundation for Lyme Disease Research. Thank you very much. I'd be happy to take questions.
Thank Professor Chiu. I think everyone agrees that the 21st century is the century of gene. So if you can handle gene, master gene, you can own everything. So this is a very good illustration of what most advanced lab can do in terms of genetic diagnosis. So I think the forum will be given to Professor Lee to moderate the panel discussion.